Day before the exam. I'm losing my voice again, which means I'm talking too much. Let's not have class, you said? Okay, I'll tell you what. I call class on Thursday. What do you say? Who says I'm not a good guy? How about extra credit? A passing grade. You're not asking for too much. Well, it's raining. I don't need anybody to wash my car, right? Say it again. So, a couple things. One, review session in here after class. I probably will finish a little early today, so that will be good. I always say that, and I don't, but I probably will finish early today. And, excuse me, get over this stupid cold. And I will videotape that. I'll get it posted as quickly as I can tonight. I should be in my office tomorrow, so if you have questions, feel free to come by. And I've written the exam, and it is, as always, similar to the format of the exam you had last time. The first section has 16 points. The second section has 51 points, and the third section has 33 points. So it tells you the breakdown of the points and approximately how many questions there are. There are 3 long answer questions in the third section, and there are 16 questions with 17 blanks in the second section. And then there's 16 questions in the first one. So pretty symmetrical exam. Question? No. Oh, I thought you had your thing. No. Okay. So, dang it. I finished talking pretty much what I wanted to say about gluconeogenesis yesterday, and so the exam material is covering through what I talked about yesterday. Everything from here forward will be on the final, and the final is comprehensive. And on the final, it's going to be a long... You're going to get tired of hearing me clear my throat. On the final exam, I do let you have a note card with you. You have to get the note card from me. Okay, that's the #1 rule. If you bring in a note card that you did not get from me, you will lose points. So you have to get the note card from me. That's the rule. All right? I'll probably say that several times between now and the exam, and even on the day of the exam, somebody's going to walk in with a different note card. Yes, ma'am? Handwritten only. That's right. So the rule is you have everything on it. You can write anything on it you want. You can use both sides. They even give you a big card. And you can do anything you want on that, but it has to be handwritten only. Now, did I tell the story in here about the student that used the different colored glasses? I haven't told the story? I love this story. Okay, it shows what ingenuity will do. So I had a student one time who says, "Do you care? Can I wear different colored glasses to the exam?" Sure, why not? Okay. So what does he do? He takes his note card and he writes as tiny as he can in red ink. Okay. And then over it, in green ink, he writes as tiny as he can. So he's got twice the density. So when he looks at it in red, he sees the green. And when he looks at it in green, he sees the red. That was clever. Very clever. I don't let anybody do that now. But I thought it was a cool idea. I really like that. Now, what you cannot do is you cannot bring in printed cards. Okay? These have to be handwritten. And you have to turn the card with your exam. And I keep the cards. So if you really have a card you really, really like, to make a photocopy of it or something, the reason I keep the cards is so that you don't pass it on to your friends. And I don't mind you passing it on to your friends, but I want your friends to benefit from making a card, just like you're going to benefit from making a card. So you pass it on to your friend next year, they're not going to benefit from that. And I think you'll see that there's a real benefit in doing the card. So that's why I require you to turn in. That's why I don't give it back to you. Okay? Not to be mean, but I just want them to have the benefit like you get the benefit. Okay. Excuse me. One thing I did not mention yesterday that will be on the final exam, it won't be on the exam tomorrow, actually concerns these metabolic control mechanisms right here. And I want to say a word about them because now, boy, you have actually seen examples of all 3 of them. So there are 3 general ways that cells have to control enzymes. Okay? One is by allosteric regulation. So you know that's when a small molecule binds to an enzyme and affects the enzyme's activity. A second control mechanism is what's known as covalent modification. You've seen examples of this where we have the zymogens. They get proteolytically cleaved, and they are activated by that proteolytic cleavage. Okay? The third example you actually saw yesterday, and that's what is listed here as genetic, and another way of saying it is control of synthesis. So, for example, the enzyme PEPCK I talked about yesterday is controlled by whether or not it's synthesized. If it's not synthesized, that's a perfect control. It can't catalyze a reaction if it's not synthesized. Okay? So these three control mechanisms cells use to handle all of the enzymes that they need. Okay? Okay. I was going to start with a song today. I'm not going to start with a song because, A, I'm not going to sing. I have it recorded on here. But I thought I'll rest my voice halfway through the lecture, and then we'll do the song then. So the song's about gluconeogenesis. So what I want to do now is, while I have a little bit of a voice, is talk about glycogen metabolism and then its regulation, which is actually really interesting. And it ties in nicely to the glycolysis and gluconeogenesis regulation that you've already seen. Well, glycogen is yet another metabolic pathway. And this metabolic pathway turns out to be very, very simple. Okay? Here's a schematic representation of glycogen. And you've already seen something like this already. Glycogen, you recall, is a polymer of glucose. It's a polymer that's used by animals, including us. And this polymer has glucoses linked in alpha-1,4 linkages. And about every 10 residues or so, there's a branch of a 1,6. So we can see, for example, that this guy has a starting point. I don't even see where the starting point is. Here's a starting point right here. Okay? There's a starting point. It goes here, and it's got a branch. It goes out to there. Goes here. It's got a branch. Goes out to there. Each of the blue items you see is the end of a chain. And as I mentioned earlier in class, glycogen is broken down starting at the ends and moving inwards. So the more ends that there are, the more glucoses can be simultaneously released from the glycogen. All right? That's a very, very big advantage of the structure of glycogen. The metabolism of glycogen is really simple. It's really, really simple. You won't find a simpler metabolic pathway. I'll show you what it is. Let's talk about breakdown first. I break down glycogen by a process called phosphorylasis, P-H-O-S, P-H-O-R, O-L-Y-S-I-S, phosphorylasis, P-H-O-S, P-H-O-R, O-L-Y-S-I-S. Phosphorylasis. Phosphorylasis sounds a little bit like hydrolysis, and it is. Hydrolysis uses water to break down things. Phosphorylasis uses phosphate to break down things. So if this were a hydrolysis reaction, this would be water right here instead of phosphate. Well, this turns out to be very simple. The phosphate participates in the breakdown, and we start with a glycogen, which is listed here as glucose with an n number of residues. And after the phosphorylasis reaction, we have glucose with n-1, and we have a glucose 1-phosphate that gets released. Now, glucose 1-phosphate excuse me, is very readily converted into glucose 6-phosphate by an enzyme known as phosphoglucomutase. Tell me the structure of the intermediate. Very good. Who said that? G1,6-phosphate, right? Mutases have that double phosphate intermediate, kind of like the 2,3-BPG. All right? So anytime you see the word mutase, that's what it has. So this guy readily makes glucose 6-phosphate. Now, why does the cell do this? Well, the cell does it because it just saved itself an ATP. If I started with plain old glucose and I tried to put a phosphate on it, what did I have to do with hexokinase? I had to use ATP to put that phosphate on, right? Right. Here, I didn't put a phosphate on. I mean, I didn't use an ATP, but I got a phosphate on there, and I've already got G6P. Is magic going on or what? Did I cheat the system? You guys say, no, because you can't cheat. It's not right, biologically anyway. So what does this tell us had to happen here? Should I save this for the final exam? Should I put it on tomorrow's exam? Well, then maybe somebody could speculate for me. How does this happen? Do you see any oxidation going on? There's no oxidation going on. It's not a bad guess, but there's no oxidation occurring here. Yeah? Well, there's no ATP here. Do you see any ATP?
Where'd the phosphate come from?
Phosphate's floating around the cell. No ATP.
What enzyme did you say?
Yeah, I haven't told you the enzyme. The name of the enzyme is called Glycogen Phosphorylase. Phosphorylase. Glycogen phosphorylase catalyzes this reaction, but it's cheating the system. It's getting a phosphate on there without ATP. There has to be something else that we're not thinking about, and there is. What are we not thinking about? Yeah?
It's done in the presence of sunlight, so a photon knocks it off.
Not a bad guess, but no, it actually occurs in your liver where there's no sunlight. Where the sun don't shine, as they say. What's that? I still didn't hear what you said. Coenzymes. Well, coenzymes don't give any energy either, but no, there's not. It's not it either.
Does it use one and then release one?
Does it do what?
Does it use one and then release one?
Well, I would still be no net gain, so no. Back here?
Is the phosphate bond being broken? Because I know that releases light.
Well, that's not a bad guess, but that's not it. But you're getting close. There's a hint. Getting close. Well, the delta G is negative. That's why it's going forwards. But why is it going forwards here? It wasn't going forwards before. There's something different. What's different, folks? What's that? It's needed, but that's not it. What?
Location.
Okay, okay. I want you guys to get this one. It'll save my voice, too. All right? I want you to get this one. Let's look at what our starting material was. What was our starting material? Okay. Was our starting material glucose? No. What was the starting material? What's the difference between glycogen and glucose? Polymers. And what do polymers have? They have bonds between the units, right? Wouldn't this tell you that there was some energy in those bonds between the units? Okay. Or are you just agreeing with me? That's exactly what it would tell you, right? There's no energy didn't come from nowhere. The energy had to come from somewhere. That tells us that there's some energy in those bonds. And the energy in those bonds are driving this process. So those alpha 1,4 bonds have a bit of energy in them. Enough energy to make this possible. We'll see that's important when we go to make those bonds. We're going to have to put some energy in to make that happen. Okay? All right. So, glycogen phosphorylase catalyzes this reaction. There's only one more thing to consider in the breakdown of glycogen. And that's here. Glycogen phosphorylase is really good at starting at the end and clipping alpha 1,4 bonds. Clips them, clips them, clips them until it gets within about 4 glucoses of a branch. And then it doesn't fit no more. So if all that glycogen phosphorylase did was that, it would only get to within about 4, and there'd be all these ends of about 4 sticking out everywhere. All the glucose would be wasted. That wouldn't be good. So cells have another enzyme that has a name. It's about this long, okay, that I'm not going to make you know. But you'll like the name of the enzyme that we give it. For short, it's called debranching enzyme. And what does debranching enzyme do? Well, it grabs 3 of these guys. This is an odd enzyme, okay? And by the way, some books call this two different enzymatic activities, but they're both in the same enzyme. All right? I don't have it listed here, but some do list it as 2 different enzyme activities. All right, so what does it do? It cuts 3 of them off. It joins them in an alpha 1,4 linkage over here. Okay? And then the very last one, it clips off, and it releases as free glucose. Now, at first, this seems like it's a very odd thing. Why does it clip 3 of them off, join them on the other, and why doesn't it just clip all 4 of them off and put them down here? That's my next question for you.
It's a different bond isn't it?
It's a different bond. So what does that tell you? What'd you say? I can't hear you. It's the same enzyme.
Can I not reattach it properly to
No. There's a different amount of energy. An alpha 1,6 bond doesn't have as much energy as an alpha 1,4 bond does. So it's not energetically favorable to take a 1,6 and make a 1,4, which is what it has to do down here. But it can take a 1,4 and make a 1,4 because they have equal amounts of energy. Make sense? Energy really matters in this consideration. So what does it do with a 1,6? It clips it off. It's not good for anything. And it makes glucose. That's the only place in glycogen breakdown that you get free glucose. The only place in glycogen breakdown where you get free glucose. Now, you've completely got the enzymes necessary to break down glycogen. Glycogen phosphorylase, phosphoglucomutase to make glucose 6-phosphate. That's not even needed for breakdown. Debranching enzyme. Bang, you're done. Okay? Very simple, right? Alrighty. That is glycogen breakdown. Let's talk about glycogen synthesis. Okay? Now, I hope, yes?
Sorry, rewinding a little.
Yeah.
The mutase, what was that one called?
The phosphoglucomutase. That converts glucose 1-phosphate to glucose 6-phosphate. It goes the other way also, as we'll see. Glucose 6 and the glucose 1. All right, so I hope I've primed you for the fact that we have to have energy to make those alpha 1,4 linkages, right? We have to have energy in order for that to happen. So this means we need to have a high energy intermediate that allows us to synthesize those bonds, all right? Well, the high energy intermediate that we use is called uridine disphosphate glucose, or UDPG. You'll call it UDPG like I do, UDPG. Now, UDPG has a glucose over here, and it has UDP over here. This molecule has a lot of energy. And the energy of this molecule is used to donate this glucose to a growing glycogen chain. Okay? You might ask, how do we make that? You might not, because then you'd probably say, well, Kevin will make us know it if we ask, right? So I'll tell you and not make you know it. How's that? How do we make UDP glucose? Well, you start with UTP. UTP is a nucleotide, right? It's used to make RNA. It's used to make UDPG. You start with glucose 1-phosphate. And how do you get glucose 1-phosphate? Well, if you have glucose 6-phosphate, you can use phosphoglucomutase, run the reaction backwards. And you got glucose 1-phosphate, right? You combine the two of them and you make UDPG. That's how you make them. So it takes a triphosphate to make this. It takes a triphosphate to make this. This tells us this guy has to have high energy, and it does. So this guy is what's used to donate the glucose to a growing glycogen chain. Here's what happens. Here's UDP glucose. Okay? UDP glucose splits out UDP. There's the glucose that we added, and you can see it going on right here. And we have an alpha 1,4 linkage when we're done. We've now grown the chain by 1. The enzyme that catalyzes this reaction is called, it's got a very complicated name, it's called glycogen synthase. Synthase. Glycogen synthase catalyzes this reaction. We're almost done, at least with this part of it. Glycogen synthase needs one other enzyme to make glycogen. What else has to happen in order for us to make glycogen?
Something to make the 1,6.
Something to make the branches. And this enzyme is very complicated. It's called branching enzyme. Branching enzyme grabs a long chain, and it transfers it down and makes an alpha-1,6 bond. Does it need ATP or not? The answer is no, it does not, because alpha-1,4 bonds have more energy. So what we've learned about energy is telling us already what's happening here, right? Branching enzyme does its thing, and we have got glycogen synthesized. Now, that wasn't complicated, was it? The answer is no, it was not complicated. All right, well, just to make it complicated, let's consider regulation. Regulation is where we get down and dirty. You've seen this figure once before, and I went over it very quickly and promised I would tell you more about it later. Now I need to tell you about this enzyme. All right? This enzyme is glycogen phosphorylase. And glycogen phosphorylase can actually exist in 4 different forms. 4 different forms. And these 4 different forms vary in whether they have a phosphate on them or don't, and whether they're in the R form or the T form. So let's consider this. All right? The T form of an enzyme, active or inactive? It's much less active, right? R form is much more active, right? All right, as we look at this guy here, we see on the top, we see the T form. On the bottom, we see the R form. Okay? How do we convert? Excuse me. All right. We also see something that's called glycogen phosphorylase B. And by the way, many books call this phosphorylase B, not glycogen phosphorylase B. I'll take either. All right. It's called phosphorylase B or it's called phosphorylase A. This is phosphorylase B. This is phosphorylase A. You can see the difference between phosphorylase B and A is 2 phosphates. Now, let's first describe how we convert between the different forms, and then we'll talk about how the different forms act. So we go from B to A by putting phosphates on. We go from A to B by taking phosphates off. Okay? Notice that those are covalent modifications. Covalent modifications interchange A and B. If we want to go from T to R, and R to T, we have to have some molecules. Okay? Now, I'll tell you that glycogen with phosphates on it, people describe as more active. We'll see why that is the case in a second. But people describe the form with phosphates on it as more active and the form without phosphates as less active. So if we were to describe a hierarchy here in terms of activity, we would say the lowest activity is probably this guy over here in the T form with no phosphates. And the most active would be over here, the A form with, the A form in the R state. Right? And these guys would be somewhere in between. Yeah?
It says inactive. You said less active
Yep.
in terms of like RT.
Yep.
Is it actually inactive?
Okay, so it's a good question. So in biology, especially as we're talking about enzyme activity, we usually talk about more active and less active, and people commonly use the terms inactive or active. Okay, but I like to use the analogy, especially with allosteric control, in terms of turning the volume down or turning the volume up. We can turn the volume way down and still have a little bit of volume, right? So that would be what I call less active. We can turn it way up, and it's more active, and that's what I'm talking about. And that's generally what's happening. Okay? All right. Now, how do we covalently modify these guys? Well, I hope you remember from what I said earlier in class that when you put a phosphate on something, you have to use an enzyme called a kinase. Okay? This guy right here, phosphorylase kinase, is in fact the kinase that does that. If I want to take the phosphate off, I have to use a phosphatase. Okay? The phosphoprotein phosphatase takes it off. Now, If I have glycogen phosphorylase B, these are both B over here, no phosphates, right? How do I convert the T form into the R form? This happens only if I have AMP. Now, why is that important? That's important because what does AMP indicate inside of a cell with respect to energy? It's a monophosphate. It's low energy. AMP will always indicate the cell is running out of energy. If the cell is running out of energy, it needs to make some energy. And so we want this guy to be active, or at least more active. What does this do when it's active? It breaks down glycogen, releases glucose, cell can have energy, right? Low energy is turning on the breakdown of glycogen.
Is that only the left side of the picture?
I'm sorry?
Is that only the left side of the picture?
That's correct. It's only on the left, only for the B form of the enzyme. Okay, I'm going through this. I will repeat this. I'm going through this kind of carefully because there's a very important point I'm going to make about it in just a minute. Okay, how do I convert the R form back to the T? Well, either ATP or glucose 6-phosphate will do this. Both of these guys are indicators of high energy. High energy is turning the enzyme essentially off, or turning the volume way down. Okay? So this makes sense. When we have low energy, we break glycogen down. When we have high energy, we turn off the breakdown. Right? Well, how about the glycogen phosphorylase A? Well, if we want to go from T to R, what does it take? Nada. Nothing. It can automatically flip from T to R without any other thing there. This is why we say that the R form, that the phosphorylated form is more active, because it's much easier for it to go from T to R.
So you'd say it's like spontaneous?
Spontaneously, it will do it as long as there is no glucose. When there's glucose, what happens? Well, when there's glucose, it flips from R back to T. And glucose is an indicator of high energy. Now, this makes a lot of sense in our liver. Let's think about this. This is going on in our liver. Our liver is a modulator of our glucose levels in our body. The liver's got to be sensing what the body's doing, right? If the liver senses that the body is needing glucose, what's it going to do? It's going to start making glucose. It's going to start breaking down glycogen. It's going to start releasing it into the bloodstream, right? The muscles start taking up this stuff. And then I stop my running. And I sit down and I catch my breath. My muscles are no longer exercising. They're going to get pretty full of glucose pretty quickly, right? So they're going to stop taking it from the bloodstream, right? So blood glucose levels are going to start rising. And when that happens, the liver is going to slow down putting glucose out, right? And if it's still breaking down glucose or glycogen inside the liver cell, what's going to happen to the concentration of glucose in the liver cell? It's going to go high. And what's high glucose going to do? It's going to flip it off. It was a bad joke. I just realized that. I just flipped you off. All right. You get it? So, this is in tune with our physiology. When our liver has caught up, this turns our liver breakdown of glycogen off. Our liver's caught up with the body's needs. It doesn't need to be dumping out anymore, so let's turn that off before we break down all of our glycogen and waste all of our energy. Very good. Okay? Well, why is this important over here? All right? Well, let's imagine that I am a muscle cell. Muscles also have glycogen, and muscles break down glycogen. I'm a muscle cell. I take off, and I was talking to somebody yesterday about going for a sprint. I was talking about jogging yesterday, but he was asking me about sprinting, so I'll talk about sprinting. Let's imagine I'm going for a sprint. I take off on a sprint, and the first thing I do is my ATP levels in my muscle cells do what? And I'm sprinting, so I'm using energy really quickly. My ATP levels are going to fall really quickly. And when my muscle cell levels start getting low on energy, they start making AMP. This immediately causes glycogen to flip into this state and start breaking, glycogen phosphorylase flip into this state and start breaking down glycogen. So why do we have to have this and this? Okay? This is the other part of the equation. All right? This left and right is controlled by hormones. Left and right is controlled by hormones. We've talked about adrenaline. We've talked about epinephrine. They're the same thing. This is the flight or fright stuff, right? It causes your body to start dumping a whole bunch of glucose into your bloodstream, right? Epinephrine activates this guy right here. It activates phosphorylase kinase. It's going to start favoring the A form, which is going to be a much more active form. Bang. Now, hormones take a few seconds to a minute to do this. All right? These things that are happening allosterically happen instantaneously. The quickest control is up and down. The slowest control is left and right. When we release epinephrine, we shift everybody to the right. If we want to activate the phosphatase and shift back to the left, that's insulin. Insulin. Okay. Now, I'm going to stop there and take some questions, and then when I've done that, I'm going to tell you a cool story. And then I'll play a song. Yes?
What did you say insulin did?
Insulin activates phosphoprotein phosphatase, moves it to the left. So your body's got some pretty elaborate controls over blood glucose levels. Very elaborate controls. And that's important because glucose is a poison. We don't want too much there. We have to have the right amount there. We have to have some there, but not too much. If we have too much there, we're in trouble. Other questions? You guys can explain this to me? Given an infinite amount of time, right? Nobody's thinking about this because we're thinking about the exam tomorrow, right? All right. I'll give you something to stimulate your thought process, all right? Well, this whole process is really important for us because, as I said, it does allow us to control the levels of glucose in our bloodstream. It won't let them get too high, and it won't let them get too low as long as we're not diabetic or something like that. If we're diabetic, we have some other considerations, okay? As long as that's happening, we're okay, all right? Well, hold on a second. We talked earlier in the term about a molecule called cyclic AMP. And we talked about it earlier relative to the cap protein in the lactose operon, if you recall in the last exam, right? Cyclic AMP, I said, was involved in the signaling process. Cyclic AMP is involved in the epinephrine cascade. There's actually a cascade of several things that have to happen to activate this enzyme. Cyclic AMP is involved in that, okay? Your cells, whenever cyclic AMP is present, that will happen with epinephrine. Epinephrine will activate and turn on the synthesis of cyclic AMP. When cyclic AMP is present, this guy gets activated and everybody moves to the right. So cyclic AMP will favor the breakdown of glycogen. Cyclic AMP will favor the breakdown of glycogen. I said it was a cool story, so it will be. Got to get there first. Well, cells, if they have a way of turning something on, they have to have a way of turning it off. What if you couldn't break down your cyclic AMP? What would happen? well, this would always be active, and you would always be putting phosphate on there. You would always have that enzyme in the phosphorylated A state, and you would always be breaking down your glycogen. You would have no glycogen. So cells have to have a way of breaking down cyclic AMP once they've made it, and they do. They have an enzyme called phosphodiesterase, Phosphodiiesterase. Phosphodiesterase. Phosphodiesterase breaks down cyclic AMP. So cyclic AMP levels go up with epinephrine, and then phosphodiesterase starts breaking it down, and you go back to normal levels. So that you don't keep this enzyme phosphorylated for too long. Because if you keep it phosphorylated for too long, you're probably going to have too much glycogen broken down, right? What if you inhibit phosphodiesterase? What's going to happen? You suppose something bad?
Deplete your energy reserves.
Okay? So if you inhibit phosphodiesterase, the consensus is something is bad. Therefore, each of you should stop drinking your coffee. Because that's what caffeine does. Caffeine inhibits the phosphodiesterase. And by the way, that's not bad for you. It seems like it would be, but it's not. It turns out that drinking coffee probably has more benefits than detriments. But the caffeine in coffee inhibits phosphodiesterase, and what it does is it favors the phosphorylated state, which tends to favor more breakdown. And when you think you get a buzz from your coffee, you really do. Your blood glucose levels are going up. You're more alert. Think about it. Your brain needs glucose, right? I need this to stay awake. I need this to think. How many people use coffee to study? I know I did. Yeah, right? It works. And it's not bad for you. There's good news, right? Next thing I'll be telling you, beer's good for you too, right? I'm still working on that one. Okay, questions about that? All right. I'm going to tell you about glycogen. I said I was going to do a song, but I'll do glycogen synthesis, which takes just a minute, and then we'll do a song. All right. No questions on this before I finish. Julia?
What did they say coffee starts to grow?
Why do they say coffee does several things, okay? So I've only told you about the glucose component of coffee. I don't know if it stunts growth or not. I can't answer that. Some people do say that. I don't know how or why that would happen. There are some nerve effects that caffeine also has that may also be slightly detrimental. But there was actually a study published in the past couple of weeks, a very large study on coffee drinkers. And there's been numerous studies now that have shown, A, that coffee has a protective effect on the liver. It's actually good for the liver, not bad for the liver. A lot of people thought it was bad for the liver. And in this study that was published a couple of weeks ago, they noted that during the study, people who drank coffee were more likely to live through the study than people who didn't. So I started drinking coffee again. Okay. Now, I don't have a figure to tell you about glycogen synthase, but I'll just briefly tell you something cool about it. Glycogen synthase is also phosphorylated and dephosphorylated. It's also phosphorylated by kinases. And the epinephrine system that phosphorylates glycogen phosphorylase also phosphorylates glycogen synthase. Now, my question to you is, what do you suppose the effect of phosphorylation has on glycogen synthase? Activate or inactivate? Why would you say deactivation?
Because the whole point of activating the but the whole point is to get glucose in your bloodstream. So if you're taking that glucose out of the bloodstream and making glycogen with it,
Excellent.
then it's not there for your body to use.
Exactly. We talked about reciprocal regulation.
Right. I think so.
That's exactly what it is. So this is another way in which reciprocal regulation is occurring. Phosphorylation of the phosphorylase favors its activation. Phosphorylation of the synthase favors its inactivation. And conversely, deactivation or dephosphorylation of the phosphorylase tends to favor its inactivation, and dephosphorylation of the synthase tends to favor its activation. That makes really good sense. What's insulin doing? Insulin is favoring dephosphorylation. We agree? What is the physiological effect insulin is doing in our body? Why does our pancreas release insulin? Absorb glucose. So this cell's getting this poison coming into it, right? What's it going to do with that poison? It's going to detoxify it. And how does it detoxify it? By making it into glycogen. The synthase is detoxifying the glucose. It's a perfect system. It's really a perfect system. Yeah?
Did you say that the synthase was phosphorylated by the phosphorylase kinase as well?
It's a different kinase, but it's the same stimulated system. That is, the epinephrine system stimulates the same thing. Okay? So this reciprocal regulation that you saw in allosteric regulation of gluconeogenesis and glycolysis works for glycogen metabolism as well. Only we see it with the covalent modification. That's how we see the reciprocal regulation occurring. So not only is insulin working to get that glucose in the cell, but it's working also to get rid of it, make it a polymer, which is not a toxic substance. Okay. That is a lot of information, and that's a place for a song, and then we'll have a review session. Today I'm not going to sing the song. I'm going to play the song for you. Okay? It's called gluconeogenesis. Sing. Oh, gluconeogenesis is so accelerated, and the rising capability is aspirated. Liver cells require it, there's no need for decay. Gluconeogenesis is so accelerating. Glucose gonna be, glucose gonna be. Also low acetate has got to turn to PEP, employing energy that comes from breaking GTP. From there it goes to make a couple phosphoglycerase, exploiting enolase and mutase catalytic trace. Oh, gluconeogenesis is liver specialty, producing sugar for the body most admirably. 6 ATPs for glucose is the needed energy. Gluconeogenesis is liver specialty. Glucose, glucose, join me. Glucose, glucose, join me. Converting phosphoglycerate to 1,3-DP3 requires a phosphate atom mutating the energy. Reduction with the lactose gives us all an NAD, and G3P's isomerized to make DHAP. Gluconeogenesis is anabolic bliss, reversing 7 mechanisms of glycolysis. To do well on the final, students have to learn all this. Gluconeogenesis is anabolic bliss. Glucose, glucose factory, glucose, glucose factory. Catalyzed reaction for sick character pieces so a fructose molecule is made with 2 phosphates in tow. And one of these gets leaked by a fructose phosphatase, unless if 2,6-PP's acting blocking pathways. Oh, gluconeogenesis, a pathway to revere, that makes a ton of glucose when it kicks into high gear. The cell's a masterminding metabolic engineer. Gluconeogenesis, a pathway to revere. Glucose, glucose, jubilee. Glucose, glucose, jubilee. From F16 to P16, that is the final phase. The enzyme catalyzing it is an isomerase. Then G16 drops phosphate and glucose it becomes inside the tiny endoplasmic reticulums. Oh, gluconeogenesis is not so very hard. I know that on the final we will not be caught off guard. Cause Harvard vessel just use a fill out index card. Gluconeogenesis is not so very hard. Gluconeogenesis is not so very hard. All right. Much better than my singing, I can tell you that. Okay. Let's see. Oop. All right, so let's break here. I'll stop the camera, catch my voice back, and we'll have a review session. How's that? Yes, sir.
I have an interview on Friday,
Uh-huh.
and I was wondering, like, if you decide